In order to understand the article, prior dietary practices and connections to a human gut microbial metacommunity alter responses to diet interventions, we have to understand the definitions of certain work. Microbiota, which are microorganisms of a particular environment; calorie restriction, which reduces calorie intake without incurring malnutrition; community, an interacting group of various species in a location; and metacommunity, a group of local communities connected through the diffuse distribution among a wide area. This experiment connects back to what we learned in class because the gut microbiota is a metacommunity that exists in a particular place, the human body, and is defined by species richness, the number of species present, and species evenness, their relative abundances. The study focuses on two groups of people in the U.S. CRON, those who practice chronic calorie restriction with optimized intake of nutrients, and Ahern, those without prescribed or self-imposed dietary restrictions. Researchers examined the effects of imposing the dietary practices on mice colonized with transplanted microbiota from the donors and the responses when the mice with different dietary practices lived together. There were fecal samples from 34 Crohn and 198 Ahern donors. The results revealed that Crohn fecal microbiota were richer and more diverse, and the OTUs are units to classify groups of closely related individuals. The phylogenetic diversity in OTUs were greater in lean than obese Ahern. The researchers then wanted to determine if the gut microbes would be affected if individuals switched in their diet. The mice on the Ahern gained 23% initial body weight but lost 14.6% at the restricted caloric level. The mice on the Crohn lost 1%, and those fed the Crohn diet at the restricted level lost 18.4% of their initial weight. Recipient communities shared similarities in their bacterial lineages that exhibited altered relative abundances in response to diets. In the co-housing part of this experiment, there were 4 groups of mice. 2 groups were colonized with Ahern 5 microbiota and the other 2 with Crohn microbiota. Ahern 1 was the target host and 1 was the control. Within the 2 Crohn groups, 1 was colonized with fecal microbiota from Crohn 1 and the other was colonized with Crohn 5. All mice were caged individually and started diets 3 days before transplantation and 10 days after switching to the opposite diet for 11 days. 21 days after colonization, all mice in all 4 groups were on Crohn. After 38 days, Crohn 1 mice were moved to Ahern target isolators. The Crohn 1 mice returned to their original cages and the Crohn 5 mice moved to the Ahern target isolator at 52 days. After 66 days, the Crohn 5 returned to their original cages. The studied mice remained in their original isolators until the end of the experiment. This was day 78. In co-housing stage 1, phylogenetic diversity increased in the Ahern target mice. In co-housing stage 2, the diversity and number of Crohn-associated OTUs increased. In stage 1, 16 OTUs from Ahern target moved to Crohn 1 mice, and in stage 2, 19 OTUs from Ahern 5 invaded Crohn 5. 4 OTUs originating in Ahern 5 mice invaded both Crohn 1 and Crohn 5. All in all, long-term dietary practices play a big role in determining the environment that the microbiota faces. Americans with unrestricted diets have less diverse fecal microbiota than those with plant-rich diets with restricted caloric intake. Therefore, placing Ahern communities into a metacommunity with Crohn enhanced the configuration of Ahern's microbiota. Thank you for watching, and we all hope you enjoyed our video.